Now, I didn't say what the QM has to be, right? Well, as long as it makes sense, it could make a good measure of like a fit. Okay? If you choose that gene set appropriately, it could save you a lot of computations. Here's an example which makes total sense in this situation. Okay? So let's consider the situation where there are a large number of candidate elements, such as gene sets, variables, such that each candidate model corresponds to a subset of something. Okay? If you say you can call them candidate elements, but basically it's a subset of something. Okay? So then, if you are in this situation, you can define a measure that fits to this case very well. Okay? So here's the definition. A measure is said to be subtractive if it can be expressed in this way. So basically, is some constant subtract something, which is a summation. Okay, and inside the summation, there's a term corresponding to each candidate element that is involved in this subset, M. Okay, so remember M is a subset of indices, right? So what you do is you take the summation over the subsets of the indices of the corresponding to the candidate elements in the subset. Okay, then you subtract that from an overall constant. Okay, so that's why it's called subtractive, because if you don't look at this, if you cover the first two terms, then it would be called additive. But because you're subtracting the sum, it is called subtractive, not additive. Yeah, because so here's another way to look at it. Every time you add an element to your model, you subtract a quantity corresponding to that element. Okay, so this kind of measure is called subtractive. Right? Then you say, what is the advantage of a subtractive measure? Well, the advantage is that if a measure is subtractive, then I can immediately find the model that minimizes this measure at each dimension. And you say, how? Well, here's how. First, I rank those candidate elements in terms of this decreasing SI. Okay, remember, there's a SI corresponding to each candidate element. Okay? So just say, suppose those candidate elements are gene sets. Okay? I have a gene set statistic, SI, corresponding to gene set I. Okay? I rank them in terms of decreasing SI. So then I can answer that question immediately. Okay? The model that minimizes QM at dimension 1 is the guy who is not ranked number 1, because it's decreasing, right? Decreasing order. Okay? The largest element is the one that minimizes QM at dimension 1. Why? Because just think about it. You have to subtract one element. Whoever is the largest, when you subtract, you get the smallest QM, right? So that answers the question of dimension 1. Then you say, how do I find the QM, the model that minimizes QM at dimension 2? Well, that's number 1, number 2, right? Because you have to subtract 2 elements. When you subtract the top 2 elements, you get the smallest reduction, okay? And so on. So with this algorithm, you find those QMs at different dimensions right away. Okay, now here's the example associated with the gene set analysis. Suppose the SI is let SI be the gene set statistic known as the gene set score, corresponding to the ith gene set. For example, SI may be the max mean statistic proposed by Efron and Tipshrani. And here's a description of how to compute the max mean. First, you obtain the positive and negative parts of each gene-wise summary statistic. So here's the mathematical definition of the positive and negative part. So then you take the means of these positive and negative parts, okay, and then you call them S bar plus and SI bar minus. Okay, so then you take the maximum of these two. That's what we call max mean. Okay, so just an example of SI. So let's just say we use this SI. Then you can define a subtractive measure QM by this earlier equation number 4, with SI being the max means. Okay, so that's just an example of the a subtractive measure. Since I have limited time, let me jump to the an example of a simulation study. Okay. So there are some practical issues that we have to solve when implement this method. But since I have limited time, I will skip those and show you this simulation study. Efron and Tipshrani, in their 2007 paper, carried out an empirical study in which the authors simulated 1,000 genes and 50 samples in each of 2 classes, control and treatment. The genes were evenly divided into 50 gene sets, with 20 genes in each gene set. The data matrix was originally generated independently from the standard normal distribution. Then the treatment effect was added according to one of the following 5 scenarios. Okay. In the first scenario, all 20 genes of gene set 1 are 0.2 unit higher in class number 2. Remember, class number 2 corresponds to treatment. In the second scenario, the first 15 genes in gene set 1 are 3.3 units higher in class 2. So then there are some other scenarios that were considered. Okay. Now we considered the same 5 scenarios in our simulation study. In Efron and Tipshrani's study, only the first gene set is of potential interest, because they only considered one situation where there's a single gene set that is differentially expressed. Now we expand their 1 gene set case to a 2 gene set case in which we duplicate the 5 scenarios to the second gene set. Also in Efron and Tipshrani's study, the genes were simulated independently. We also considered a case where the genes are correlated with equal correlation that is equal to 0.1. This describes how you generate the correlations. We basically use the mixed effects model to generate data with the correlation, which I have to skip. Okay, so then we compare the performance of IF. IF means invisible fence, with GSA in gene set identification. In the Efron and Tipshrani simulation study, the authors showed that the max mean, the one I discussed earlier, has the best overall performance as compared with other methods. So therefore, our comparison focused on the best performer of GSA, that is the max mean. In addition to the 1 gene set and 2 gene set cases, each with 5 scenarios listed above, the simulation comparison also includes a case where there's no gene set that is potentially of interest. Okay, so we consider 3 cases: no gene sets, 1 gene set, and 2 gene sets. The number of permutations for GSA is 200. That's the number that Efron and Tipshrani used. The number, the invisible fence requires a bootstrap procedure in order to get the empirical probability. Remember that empirical probability actually was obtained by bootstrapping. So that the numbers bootstrap samples is used that are used as is 100. Okay, now here's the first set of comparisons. Is on the probability of correct identification of truth positive. And what it means is that you identify exactly those gene sets that are differentially expressed, no more and no less. So that's called truth positive.